All right, can everybody see that?
Yes, we can.
Excellent. All right. So hello, everybody. My name is Austin Bruce. I'm a third-year doctoral student at the University of Iowa in the neuroscience graduate program. My mentor is Dr. Nandakumar Narayanan, and the title of the talk is going to be Experience-Dependent Remapping of Temporal Encoding by Striatal Ensembles. All right, so I think we can all appreciate how precise temporal control of movement is key to our success in everyday life. Ethologically, it's the foundation of predatory and escape behaviors, but there are a number of dimensions upon which we must precisely control our actions in time throughout the day. A deep brain structure which allows us to precisely engage motor responses exactly when they are needed is the striatum. And it's known that patterns of striatal activity dynamically change as we acquire new skills or form new habits. The research we're going to talk about today aims to elucidate how striatal circuits adapt as we learn novel motor tasks for which the time we respond is the critical factor. So I think we might have some intuition about the organization of the basal ganglia, but we're just super briefly going to go over sort of a 40,000-foot view. So the striatum forms a critical node in the action selection circuit. So by integrating dense glutamatergic input from the cerebral cortex, particularly layer 5 neurons, the striatum exhibits indirect control over thalamocortical circuits, which project to layer 4 of the cortex through a chain of downstream sequentially organized output structures. So here's a dye stain of a medium-spiny neuron on the right here, and here's sort of a very simplified diagram of the direct pathway of the basal ganglia. So the idea that medium-spiny neurons might encode time via ramping behavior, that's progressive monotonic increases or decreases in firing rates, preceding salient temporal events in time, has been around probably for the last maybe 2.5 decades. Here's a figure from one of the first publications that demonstrated this phenomenon incredibly well. So these are some raster plots from a peak interval task in which rodents are rewarded for responding in either 10 or 40 seconds. So on the top, we have trial by trial raster plots. The y-axis are trials, the x-axis is time. Each tick you see is a given time this neuron fired. There's spike bend histograms, that's just counts of every time the neuron fired on the bottom. And then these black traces correspond with response rate. And what we can grossly appreciate here is that there's sort of an uptick in activity preceding the 10-second target for these two cells, and an uptick preceding the 40-second target in these cells. So this might be one mechanism through which striatal circuits encode time. So about 4 years ago, my colleague Eric Emmons replicated this result and expanded upon it with a new task. So here's a trace from a single medium-spiny neuron in a rat that's been trained to respond at either 12 seconds, here in this dark green raster and this dark green trace, or at 3 seconds in this light green raster and this light green trace. And what I want you to appreciate is this neuron is able to dynamically modify the slope and its change in firing rate over time to exceed a certain threshold at the target time. And he also showed that this dynamic is dependent upon similar processes taking place in the medial frontal cortex, but that's a discussion for later, I think. He also showed that this phenomenon is largely independent of motor activity. So this is a representation of the ensembles he recorded. He found about 45% of the neurons he saw in the dorsal medial striatum express this time-dependent ramping behavior phenotype. However, he also found a population that were responsive when the animals made motor responses at the appropriate time, right? The lever press. However, there was a significant overlap between these populations, accounting for about a quarter of the ensemble. So, just taking a little bit of a detour here into the motor field and the habit field, it's been demonstrated that medium-spiny neurons become more sensitive to the same actions as animals undergo training and overtraining. So this is a publication from back in 2003, where Tara Barnes, who was working with Anne Grabiel's group at that time, she trained rats to run a T-maze over the course of over the course of 21 days. And so how this works is the rat starts off in this initial corridor. The gate is open when the trial starts. The rat traverses through the corridor, and then one of these arms contains a food reward. Which arm that's going to be is indicated by one of two tones here. And what she found was that individual medium-spiny neurons in the striatum became remarkably more selective for individual action components of this task as the animals underwent training. So on the top panel here, we have a neuron which is sensitive to locomotion onset. And what we can generally appreciate is that, so time zero, let me make that clear, time zero here is locomotion onset. Here's some spike bin histograms and some raster plots. And what we can appreciate is there's a greater likelihood that this neuron generates an action potential with training on this task over this period of 6 days. Here on the bottom panel is a neuron which is sensitive to turn onset. And so although the motor actions couldn't be more similar, we noticed that the response profiles change over this time. So, just to briefly set us up for the data we're going to talk about, we've talked about how striatal medium-spiny neurons may encode temporal intervals via time-dependent ramping behavior. It's a process which is largely dissociable from motor activity. We've also talked about how medium-spiny neurons actively change their response profiles in response to discrete motor actions as animals undergo task training. So, a very simple question could follow from this is, does time-dependent ramping behavior follow the same principles? That is, does it become more prevalent with time? And kind of zooming out, we could repose this question, or can we say, does this non-motor activity profile obey the same principles which Barnes illustrated in her paper back in 2003? So to investigate this question, we trained a cohort of 6 mice on an interval timing switch task. This is based upon a task design that Fuat Balche put out in a paper around, I think it was 2008. So credit goes to my colleague Matt Weber for designing this this wonderful task schematic here. So let me just walk you through this. So in this task, mice are presented with 2 different trial types. In half of the trials, so-called short trials, they will be rewarded for their first response at 1 of 2 nose pokes and an operant box at 6 seconds, right? So here's a raster of behavior. These are individual nose pokes. The first response after 6 seconds, this mouse is rewarded for. However, in the other half of trials, mice are rewarded for their first response after 18 seconds. So here it's the right nose poke, which is the correct one. After making some initial responses at the left nose poke, the animal notices that the cues have exceeded 6 seconds and switches over to the other nose poke and is rewarded for that. All right, does that make sense? Let's watch a video to kind of get everybody on the same page here. All right, so that was a long trial. The mouse initially interacted with the short latency nose poke and then switched to the long latency nose poke and was rewarded for the first response at 18 seconds. Now, the analyses that I'm going to present to you today are focused on these long trials because there's a number of interesting temporal events. Firstly, we have short latency response at 6 seconds. We have the long latency target at 18 seconds. And we have when the animal decides that 6 seconds has elapsed, I had better switch from one response port to the other. So here's just kind of a brief schematic of my experimental timeline. So initially, mice are implanted in the dorsomedial striatum with 4x4 multi-electrode recording arrays. After a recovery period, they're trained on this task for 14 days. After that, they're habituated to our recording procedures. That is, we plug them in as if we're going to run an experiment. We let them explore the chamber just to get used to the dynamics of moving around while tethered. And then we run 10 sequential days of tethered behavior. Now we're going to focus on 2 time points here. The first time point I'm going to show you is so-called early training. That refers to the first time the mouse got more than 5 correct trials. The second time point I'm going to show you is so-called experience. That's the last time we recorded from that animal, day 10. So as I mentioned, we implanted 4x4 multi-electrode arrays into the posterior medial aspect of the dorsal striatum. There's a lot of, I guess, commentary on the functional roles of different subunits within the striatum, whether it's dorsomedial striatum or dorsolateral striatum. So we thought it might be an interesting idea to incorporate histological techniques that allow for independent verification and reconstruction of array orientation. We did this by running a current through what should have been the posterolateral-most electrode 16 in this case, then staining the tissue with Prussian blue, and the counter stain here is nuclear fast red. So, armed with this lesion data and the array map, you can get a better idea of where any given pin is. So let's look at our behavior. So this is sort of a 40,000-foot view of what's going on. Here are raster plots comparing the early versus the experience session. Short latency responses are plotted in gray. Long latency responses are plotted in black. The switch departure time is plotted in green, and the arrival at the contralateral nose poke is plotted in blue. And so we can generally appreciate that mice are transitioning to becoming a little bit more active in the later trials, although number of trials per se didn't necessarily have a consistent change between all animals in both states. Now, looking at some bend response histograms, and when the animals as a group were likely to interact with the short or long latency response ports, we find these two partially overlapping distributions. So short latency responses are plotted here in gray, long latency in black. We see the peaks of these distributions are discrete, but there's a huge overlap in the middle that doesn't go away with experience. Additionally, we found that average switch time, that is the departure from the short latency nose poke and traversing to the long latency nose poke, didn't consistently change between all animals. Maybe there's a convergence towards a sort of 10-second mean, but there's no significant effect here. Additionally, there was no consistent effect of trial to trial variation, either going up or going down with further training. However, one metric that did improve was the proportion of correct responses. That is, if I got a short latency trial, I responded at the appropriate port at 6 seconds, or if I got a long latency trial, I responded at the appropriate port at 18 seconds. And so that increased from about 60 to 80% in the group overall. Now, turning to our neural analysis, we saw large populations of neurons which expressed time-dependent ramping or expressed this sort of nose poke sensitive response profiles. So here's an example of a response sensitive neuron. Time 0 is a beam break on either the short or long latency nose poke. And here's an example of a cell that is remarkably sensitive to this event, right? So here we can see a transient peak in activity preceding 0 seconds, some transient inhibition, and a further peak in activity. And it's remarkably consistent across all trials. Now, as I mentioned, we saw large populations of time-dependent ramping neurons. So here's an example of that. Here's a cell where we have some trial initiation related activity, but as the trial progresses, we see this progressive increase in firing rate until the trial ends. So this is an example of a cell expressing time-dependent ramping behavior. Now, to quantify these observations, we used a generalized linear model approach where we asked the basic question: is there a relationship between firing rate on one hand and either time in trial or response on the other hand? So here's sort of a graphical representation of what we found in our ensembles. So 80 medium-spiny neurons early, 81 medium-spiny neurons late, somewhat balanced between all 6 animals. We found that there were large populations of both ramping and response-sensitive neurons, but there was a large population that overlapped. Comparing between the two training sessions, we found a large increase in both the ramping and response populations, largely driven by a massive increase over 2-fold in this multiplex category. So doing both time-dependent ramping and response sensitivity. So that's kind of interesting. We've expanded Tarabaran's initial finding with respect to motor activity to encompass time-dependent ramping. So beyond just ramping becoming more prevalent with time, there might be higher order changes that accompany training. That is, ramps may become more precise, they may span greater periods of time, or they might be less variable in slope. And there's conceivably many other schema through which neurons can encode the passage of time that might exist. So, human beings and neuroscience graduate students in particular are less adept at quantifying complex multifactorial changes like these, but machine learning algorithms are exceedingly efficient at doing just that. So in this case, we turn specifically to naive Bayes classifiers. So a general sort of view of how they work is based upon a training data set composed of binned and labeled Z-scored firing rates from a subset of trials across the ensemble of neurons. They develop a very simple sort of schema of how firing rates should relate to time. And then based upon a test data set composed of other untrained trials, you ask the classifier to assign given bins of activity to the moment they occurred in trial. Sorry, didn't mean to jump ahead there, but I did. Why do we specifically choose naive Bayes classifiers? Well, naive Bayes classifiers assume strong independence between all variables in your training data set, and therefore any performance on their part is largely due to the quality of the training data set. So if it decodes time well, it's because temporal signal in your training set is better. If it decodes time poorly, it's because the quality of the training set is not great. So here's sort of a graphical representation of what we found. On the x-axis between these two sessions is observed time. That's the actual time in trial a given bin of neural activity was sourced from. On the y-axis is your predicted time. That's the time the naive Bayes classifier chose as the likely time point of origin for that bin of activity. The probability of selection of any given bin is indicated by this color bar. So dark colors indicate it's unlikely, light colors indicate that it's likely. With perfect performance, we'd have an x equals negative y line running from top left to bottom right. But I think looking at this, we can appreciate that performance is a little bit more variable in the early versus the experienced session. So there's more variability, and then we get this nice, little bit tighter line here. So the way that we rather evaluate our performance is through computing variance explained, R squared, between observed time and predicted time between the two training data sets. And what we find when we compare the early versus experienced session is that decoder performance increases with the experienced training set. Now, there's sort of a negative control next to either of these violin plots, that's shuffled data. That's the same training set was used, but the bins were randomly aligned in time. So there's no real temporal signal there. Something we do not expect to work doesn't work, and that's reassuring in a sense. Now, digging a little bit deeper, when we comprise the training set exclusively of ramping plus response or ramping cells, we found that the classifier performance was much better than when they were trained exclusively on response modulated or non-modulated neurons. So that suggests that perhaps there's a causal relationship between the increased performance we see in the experience session and the increased prevalence of time-dependent ramping activity. Now, this sort of dovetails with a finding that my colleague Eric made back in August of this year. This paper was published in Cerebral Cortex back in August, I'm pretty sure, where he found that temporal decoding improved for the dorsal medial striatum, specifically over the course of 3 days as animals were trained on a novel 3-second interval timing test. So in a sense, we've replicated this result in a different paradigm over a longer period of time. Finally, I asked the question whether or not it would be possible to predict whether or not a given trial was an error or not. That is, the terminal nose poke was at the inappropriate side based upon neural activity. So once again, relying on a generalized linear model approach, predicting firing rate on the basis of, or asking whether or not there's a relationship between firing rate and time, and whether or not that trial was an error, we found that a subpopulation of neurons, about a quarter in the early or experienced session, had significant differences in activity profiles on correct versus error trials. So here's an example of that. The correct trials are plotted here in black. Here's an average trace here. The error trials from the same cell on the same session are plotted in red. And we can appreciate that there are some differences. We see this density of activity preceding the 6-second target. However, that's not there in the error trials. And notably, this is not a difference in time-dependent ramping. Whether or not a cell expressed time-dependent ramping didn't predict the success of any given trial. So just to conclude, we've talked a little bit about how striatal encoding of time may adapt as animals gain task experience. Firstly, the number of MSNs exhibiting time-dependent ramping activity appears to increase with extensive training. We saw that temporal decoding dramatically improved with experience, and we believe that this is a process that is driven largely by an increased prevalence of ramping activity. Finally, it appeared that a subpopulation of medium-spiny neurons expressed different activity profiles on both correct or error trials. And this is something that I want to dig into a little bit deeper. So, I want to acknowledge everyone who's been a part of this journey. Specifically, I want to thank Matt Weber, Mayu Oya, and Rachel Volkman for helping me out with these experiments. I want to thank Eric for being somewhat of a mentor figure, and of course, I want to thank my mentor and our funding sources for giving me the opportunity. So, if anyone has any questions, I would be happy to address them. Oh, sorry.
Awesome. Thank you so much. You're good. Thank you so much, Austin. If anyone has a question, they can raise their hand. I see. Oh yeah,
I see one from Nathaniel.
Hi. Yeah. Thanks for that. I'm sorry if I missed this, but I was wondering if you could clarify how you define ramping activity, and specifically, like if a neuron ramped up in firing for like 8 seconds, but not the entire 18-second interval, is that considered ramping activity?
Well, to do that, we basically used a generalized linear model, which is essentially a special case of a regression. We asked over the entire trial, is there a relationship between firing rate and time in trial? So yeah, it's possible that we can miss some transient ramps earlier, or maybe just preceding reward delivery, but typically it's sensitive to trial spanning ramps. And this is sort of the phenotype that this method picks up, but it's also sensitive to other things. Let me see if I can pull up another example. Sorry. So it was also sensitive to negative ramps. Here's another example of a phenotype. We have a ramp to 6 seconds, then this decrease in activity preceding a trial end. So this might be another like encoding schema that's present within the ensemble. I'm sorry, does that answer your question?
Yeah, that's really interesting. Thanks.
Any other questions for Austin?
I think there might be some in the chat.
If they are, I don't see them, okay, unless I'm missing it.
I have a question.
Yeah, go ahead, please.
All right, so this was really interesting. Thanks. Sorry if I missed this, but you're saying that you're recorded from almost 80 medium-spiny neurons in a striatum. Do you differentiate between D1 and D2 medium-spiny neurons?
I do, but this data set didn't.
Okay.
So this data set didn't explore that, but hope to revisit it in the future.
All right. Okay. Thanks.
I have a question. It's Peter Balsam.
Please.
looking at the tracings that you showed us of individual trials, kind of looks like maybe ramping is the result of averaging over trials rather than what happens on individual trials. What do you think?
Well, so I should have made this clear. So our generalized linear model looks at relationship trial by trial, right? And you know, I agree with you that let's just pull up a trace really quick so we can illustrate this point clearly. So you know, if we were doing it on the basis of averages or you know, something similar, if we had a density of spikes here, you know, it might, in a couple trials, it might give the impression of ramping where it doesn't actually exist. However, I don't think that was a significant problem for us. But yeah, I see your point, and that's why we took it into account when we designed the model initially.
Awesome. I think we have time for maybe one more question. But if that's it, then thank you so much, Austin. That was really interesting. And if you could just stop sharing your screen, then we can get set up with Dr. Jonathan Cannon. Awesome. So our next speaker is Jonathan Cannon, at postdoc at MIT. So he's a postdoc in the Cine Lab, where he is doing behavioral and EEG research that takes a predictive processing perspective on autism. His graduate training was in dynamical systems, modeling of neural systems with Dr. Nancy Kopel. By night, he performed semi-professionally as a traditional fiddler and guitarist. His life of music drew him to Dr. Annie Patel, with whom he has been working closely for several years to understand the neurophysiological mechanisms underlying the human faculty of rhythm. So thank you, Jonathan, and you can start sharing your screen.
Unmuted. Fabulous. Okay. Let me start the show. Okay, so what I have to share today, thank you so much for an excellent presentation, Austin. What I'm sharing today is not experimental work, but instead some synthesis work. And I'm trying to put it under the umbrella of the shared predictive roots of motor control and beat-based timing. And we'll talk a little bit about striatum as well. So first of all, what am I talking about with beat-based timing? I'm going to give some background on what I'm referring to here. And then with the rest of the talk, I'm going to talk about a set of hypotheses that Ani Patel and I put together on the roles of structures in the brain that are traditionally considered motor structures in beat-based timing. And what we're trying to do is review known motor functions and go to the motor literature and try to extrapolate from that and be as specific as we can about what we think those motor structures are doing in beat-based timing. And then at the end of the presentation, I want to reframe those hypotheses in terms of an idea called active inference that I'll introduce you to, which positions those roles of those motor structures in beat-based timing not so much as co-opting a system that is generally used for moving our bodies, but instead as really the primary functions of those motor structures. So from the beginning, beat-based timing, we're talking about as the when a rhythmic stimulus gives rise to the identification and maintenance of an underlying periodic pulse. And that sense of a pulse can then be used to make timing judgments, which is where timing comes in. That includes tracking the instantaneous phase of the cycle of the pulse, and as well as the tempo, which is preferred around between 300 and 1,000 milliseconds. And so I'll give a quick example of that here. We've got a complex rhythm that will induce the sense of an underlying pulse. And so if you're to tap your finger along with this, you might tap somewhere around here. And so what these arrows are indicating is that while some of these pulses line up with the clicks, others do not. There are times that a pulse is experienced, but there's no click. There are times that clicks occur, but there's no pulse. In other words, this is an internal construct that is meant to fit the rhythm that we perceive, but is not physically present in it. Once this pulse has been induced, that inferred phase and tempo can be used for perceptual tasks like early or late timing judgments, or for enhancing perception at specific phases, or can be used behaviorally in these examples, including precise motor entrainment, which humans are remarkably good at. We can tap along with the beat very accurately and step along with the beat as well. So this is beat-based timing, and some excellent fMRI work over the years has looked at what brain areas are important for beat-based timing, and in particular, we'll focus on covert beat-based timing when entrainment is not involved. So first comes up the supplementary motor area, which is the medial surface of the cortex in the premotor region. There are two different sub-areas here, the SMA and pre-SMA. I'm not going to draw some distinctions between them in this talk, but there are some important distinctions to be made. So apologies to people who study that more closely. But I think most of what we'll be talking about here will be SMA proper. The basal ganglia, especially the putamen, which is often considered a primarily motor structure in the deep brain nuclei of the basal ganglia. And also lighting up, those are the two I'll be focusing on today. But the lateral premotor cortex shows up, auditory and parietal cortices show up, which together with premotor cortex are often called the dorsal auditory pathway. Cerebellum shows up sometimes, but it seems to be more involved in physical entrainment or in rhythms that are not periodic, but are produced from memory instead. So I won't be focusing on that very much. So some important notes here in how this information has come together. A lot of this region-specific human data is from fMRI, which means that we don't get to see the actual dynamics from one beat to the next within the regions of the brain. And when we do get to see that, it's in animal models who don't seem to have the same relationship with the auditory beat as humans do. Humans are much more accurate in entraining movements and seem to get a lot more out of beat-based stimuli than animals do. So that's why this area is ripe for hypothesis, and not a whole lot is known yet. So the big question, what are these motor areas actually doing during beat-based timing? It's been widely hypothesized that they play some kind of active or causal role in timing. And more recently, it's been proposed that simulated actions are being used to time intervals, these periods from one pulse to the next. But this is somewhat underspecified. It's hard to say exactly what a simulated action means physiologically. And so my approach at this point is to try to extrapolate from the things that we do seem to know that those structures are doing during motor control, and then try to understand whether those particular functions generalize into beat-based timing. So we're going to start with the supplementary motor area. A recent set of papers from primate SMA have shown they've looked at the geometry of firing rate trajectories, which is to say you take a whole lot of different neurons, you graph all of their firing rates over time in a big high-dimensional space, and you look at the shapes that makes. The shapes have been observed over a couple of different papers to make shapes that are consistent with the idea of tracking progress through a periodic action. So here's an example where a monkey is turning a crank in a circle. This is from the Churchland lab. And what you see is these firing rates in SMA making loops, one for each of these crank turns, but you also see some change in a different dimension in that space. And so we end up with sort of a spiral here. And the idea that's been put forward for what all that means is that SMA is tracking progress through a single repetition in the form of a loop, and more broadly tracking progress through the whole movement as it sort of spirals through the space here. And this is 3 principal components. This is just a way of visualizing very high dimensional data in a lower dimensional space. So here's another study that had similar results from the Merchant lab. There were results in which monkeys that are periodically tapping their finger will see in supplementary motor area these similar loops. And in this case, an important aspect of this is that part of this time, the monkey is just waiting with his finger above the tap pad. And so that indicates that this is not really closely related to the actual movement itself, but instead is tracking the progress of the movement, which may include some motionless time. It's a more abstract representation of the movement. And a third paper from the Jazairi lab showed, in addition to behavioral progress tracking activity in SMA, that the time course of the firing rates in SMA seem to be created through local interactions between neurons in SMA. In other words, we've got some autonomous dynamics here. It's not being driven over time by some upstream region, though it may be modulated by input from outside regions. So that's the picture that's been emerging from these motor studies in animals in SMA. Looking at a totally different body of SMA literature, it's been known for some time that SMA is involved in enhancing and suppressing self-generated sounds in humans. And so these old results show that SMA, I mean, this seems somewhat contradictory, enhancing and suppressing, but I think bringing those together, basically SMA is involved in predicting when self-generated sounds are going to happen so that that information can be used by the perceptual system in whatever way is necessary. And a set of prominent models suggest that self-generated sound prediction is moderated by lateral premotor cortex, which contains these forward models of how a particular action should create sound. And so bringing those ideas together, we've got this sort of simple picture in which the phase of a action in SMA informs the premotor cortex of where we are in that particular action process. And then the premotor cortex, based on that, can tell the auditory pathways involved what to expect to hear at that particular phase of movement. So I'll be building on this picture. So taking this whole idea from both of those sources of data about SMA and trying to fit it into beat-based timing, we can actually just use exactly the same picture and say, well, suppose that SMA state tracks the phase through an anticipated interbeat interval, rather than through a behavioral process, but just through an external process. Then that phase can inform the premotor cortex, which in this case is containing a forward model, not of the sound expected to be produced by an action, but the sound expected to be produced by whatever this process is out in the world. And that can then send that information about what to expect at that particular phase to auditory processing, which can then either enhance perception or suppress depending on the specific needs. So I think all of this fits together fairly nicely as an idea of the possible role of SMA in tracking the beat. And those loops really make SMA look a lot like an oscillator. This has been a sort of classic framework for understanding beat-based timing. But I don't think that's actually what's going on. I think that an oscillator, well, we can talk a little bit more about the distinction. And I think zooming in on basal ganglia will help to make that distinction between what SMA is doing and the oscillator models. So the classical understanding of the role of basal ganglia in movement is that the direct pathway through basal ganglia, which comes from cortical regions, a broad area of cortex, into the striatum. Striatum inhibits another nucleus, globus pallidus, which inhibits the thalamus. And the thalamus then excites these various different motor regions, including primary motor cortex, premotor cortex, supplementary motor area. And that's understood to be to carry disinhibitory signals that can use this cortical information to disinhibit a specific appropriate movement plan that can go next. And the cortical signals that can feed into that include sensory context. In other words, based on what I see and hear, that can help me select an appropriate movement plan for this moment. But also my own current motor state. And that allows me to sense what I'm doing at this moment, where I am in that action, and use that information to select and disinhibit the next action. And this gives basal ganglia this central role in creating sequences of movements. This has been studied pretty extensively in rodents, including in this paper I'm citing below. Okay, so let's look at the interaction of the supplementary motor area and basal ganglia during periodic action. This has not been mapped out in exactly this way. I'm trying to interpolate from existing results. So it's not exactly new data or new ideas that I'm presenting here, but it's not quite old ones either. Just trying to sort of construct a picture of what's happening. So SMA, based on this primate data, is generating these short loop trajectories that represent progress through a repeated movement. And in the meantime, basal ganglia is laying down the track for the next loop that SMA is going to make. And so this is saying that the basal ganglia's modulation of SMA is determining exactly what loop it's going to make, where that trajectory is going to go. But the SMA is then generating that trajectory of firing rates through its own internal interactions. And so, right, basal ganglia input is shaping the conditions in SMA, which determine the dynamics over the next interval. And that's consistent with the picture from the Jazayeri lab that I mentioned before. And so here's that same picture of a monkey turning a crank from the Churchland lab. And I'm going to try to animate this and show what role the basal ganglia may be playing here. So basal ganglia disinhibits a particular set of cells in SMA, which effectively give it the command to make a loop. It makes a loop of firing rate trajectories. And as it reaches the end of that loop, basal ganglia then issues or disinhibits another set of cells, which may be overlapping, maybe slightly different, telling it to make another loop. And it does that, and so on. And so basal ganglia is playing this role of sort of laying down the immediate future for the supplementary motor area. So our hypothesis about the role of, about the interaction of SMA and basal ganglia in beat-based timing is that basal ganglia is laying down the track for the next SMA loop as it tracks progress from one beat to the next. And in this particular case, we also could see that the basal ganglia input changing from one cycle to the next could allow for a hierarchical representation of beats. So in the same way that the basal ganglia's input can make one loop of a monkey turning the crank slightly offset from the one before it and slightly offset the next one, giving us both a phase representation and this higher order representation of which turn we're on. The basal ganglia's input changing from one loop to the next would allow for a hierarchical representation of the 4 beats in a 4/4 measure, 1, 2, 3, 4. We might return to the beginning. Now this seems a little fantastical and far-fetched, but actually there's some recent results showing hierarchical representations of repeated movements in basal ganglia in rodents. And so they were pressing on a pad left, left, right, right. And the activity in basal ganglia seems to contain this type of hierarchical representation of those actions. So we think this is pretty plausible. All right, so it's not an oscillator then. It's not actually self-sustaining an oscillation, but instead it's getting coached from basal ganglia above that's sort of laying down these tracks. And one way to think about this is rather than an oscillation, keeping the beat is a series of predictions, one after another. And in this case, the predicted interval is often, but doesn't need to be identical. And this can depend on context. So what strings these predictions together from one to the next? What makes this periodic and not just a disconnected series of one prediction, another prediction? And the hypothesis for this is that dopamine plays an important role. So let's talk about that. So dopamine is often thought of as reward prediction error these days. But before dopamine was reward prediction error, there was a whole literature that was attributing it a role in action initiation, that pulses of dopamine initiated action. And actually recent data has really brought that back into the fore. So these 3 papers here, 2016, 2018, 2019, all using newer, more temporally precise recording methods have shown that pulses of dopamine seem to be present and even necessary and sufficient to initiate actions. And yeah, so this is distinct from the ventral striatum dopamine, which is more closely associated with reward. And this is in the dorsal striatum. And these pulses accompany external action cues, go cues, or can be generated internally. And those internal cues are likely timed by the supplementary motor area. And so this sort of brings us around the loop again. Basal ganglia is telling supplementary motor area what to do, and supplementary motor area is helping give these cues for basal ganglia to disinhibit actions. So our hypothesis here is following that motor literature. We're hypothesizing that sounds on the beat provide a dopaminergic pulse for initiating a new loop in SMA, but that silence on the beat would then necessitate an internal pulse of dopamine generated by the state of SMA. And so with strong dopaminergic signaling from SMA, you'd be able to then continue that beat in the absence of input. Every SMA loop would have to issue a new dopaminergic pulse to dorsal striatum to tell it to go and run the next one. And so that's sort of this balance of external influence on the beat, but also the possibility of continuing it internally. So that takes us through these hypotheses. And now with the rest of my time, I want to reframe these hypotheses in terms of active inference. So to introduce active inference, we need to start with predictive coding. This is a prominent idea that helps understand sort of the brain at large, and in particular, the hierarchy of cortical regions. And this idea, which is fairly well supported by experimental data now, suggests that higher areas of cortex represent inferences about the state of the world, things that may not be directly observable, that descending connections from those areas carry predictions based on those inferences, and that ascending connections from the lower sensory regions, like the primary visual cortex, report not so much just what they're sensing, but errors in those predictions. So only if you see something unexpected do you get a strong signal from primary visual cortex moving up the hierarchy. And the prediction errors that are being signaled in that way lead to the adjustment of inferences and predictions at every level of cortex. So what is active inference? So this is applying the same idea to the motor control. So it's been observed that from physiological study of motor cortex that the laminar and topographic and physiological properties of the motor system suggest that the same principle is in play, in which descending connections carry predictions, although in this case, they're proprioceptive predictions. They're predictions for what I will experience as I move, for what it will feel like to be in a certain place, moving at a certain speed. And then those predictions, unlike sensory predictions, are actually fulfilled by motor neurons. So our motor effectors are self-fulfilling the predictions we make about where we're going to move. In other words, movement is about expecting to move, not about commanding parts of our body to move, which I think lines up a lot better with our subjective experience of movement. And then ascending connections are also conveying prediction error. For instance, if I'm moving and my hand hits a table unexpectedly, I'm going to get some proprioceptive prediction errors. I expected my hand to be down there, and instead it's up here. And those errors are then used to update my proprioceptive expectations, which at higher levels of cortex correspond to motor plans. And so here's a big picture we don't need to parse completely, but it's from this excellent paper by Adam, Shipp, and Friston, in which it shows on the left, this is sort of the prevailing ideas about motor control, which have inverse models and forward models, and the motor cortex is sending a forward model of what it's going to do to sensory areas. But rather than talking about this as forward models and inverse models, we can see the same set of connections as relaying predictions and ascending prediction errors. So it's almost exactly the same set of connections. It's understanding them a little bit differently, and the predictions about what you would observe experimentally are slightly different. But it's mostly just a very different framework for thinking about the motor commands and motor feedback. And so if we think about motor control in this way, then we can think about how the motor system might use auditory feedback from movement. So here's the motor system, primary motor cortex sending predictions about what to expect to propriocept to these motor effectors, which then move to correct those prediction errors. And any additional prediction errors are sent back up the hierarchy. How wrong were those predictions? So SMA shows up here, and it's representing a real-time inference of the phase of this movement. And so it's making that prediction to M1, and then errors in M1 can update SMA and say, oh, we're actually not as far through this movement as we thought we were, for instance, and can then update the estimate of the phase of that repeated movement. And then that predicted phase is also setting the time course for motion because its predictions are getting fulfilled by motor effectors. So this is sort of bringing together the idea of SMA as motor planning and SMA as a inference of the phase of the current movement. And then SMA can also inform the predictions of auditory motor feedback. And so SMA is sending that same information about the phase of the movement to premotor cortex, which says what it expects to hear now to these auditory pathways. And they're forwarding back up the measures of prediction error. How wrong were those predictions? Which can also be used to update the phase in SMA. So in isochronous finger tapping, this might look like this. SMA is sending that information that we are very close to the end of a tap cycle to both of these regions. And in motor cortex, that then sends the prediction that we will feel our finger lowering. Motor effectors fulfill that prediction. And if there's any error, that gets forwarded back up to SMA, which can then adjust its sense of phase if it's not quite aligned with where it expects to be. And on the left, we now have this the same signal is getting used by premotor cortex to predict that we're going to hear a tap really soon. And so if we don't hear a tap, that error can then get forwarded back up to SMA to adjust our phase of our tapping. And if we're listening to a drum track, well, we might not have any specific proprioceptive expectations for what we're going to be doing when we listen to a drum track. And so this area for now is grayed out, especially if we're instructed to listen covertly, in which case we have no proprioceptive expectations at all, or the expectation that we'll sit still. But we do have auditory expectations, and that's how this left side of the diagram can function. SMA says that we're nearing the beat. PMC says to expect a kick drum. The auditory pathway says, oh, didn't hear a kick drum. And premotor cortex says, okay, then I guess the SMA phase is a little bit ahead and can drag it back. And so, yes, in this case, we've got covert auditory active inference, which is really just a fancy way of saying auditory predictive processing, which naturally uses the motor system. And I've got a preprint on this particular aspect of this system that looks at this as a formal inference problem and solves that problem and makes some fun, it reproduces some cool experimental results and makes some fun predictions. It's called Pipit Phase Inference from Point Process Event Timing, and I've got that up on bioRxiv. And bringing the basal ganglia into this at last, it's disinhibiting predictions about the next chunk of time in all of these different regions. So it's predicting what I would expect to propriocept during this next chunk, informing these motor cortical proprioceptive predictions that are going down to the motor effectors. It's informing PMC of what to expect to hear during the next chunk of time. And it's informing the supplementary motor area of how long the next chunk will take. And what's really fun about this is that if this is our mechanism for beat perception, then we don't necessarily have to have the same beat duration over and over again. It can depend on context, which would allow for sequences of different beat durations, like you see in 7/8 time in Eastern European traditional music. You know, dum 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 dum. So coming back to the big question, why do humans use motor areas for precisely timed auditory prediction at all? And based on this active inference picture, I'd say that the answer is because the motor system is essentially a system for dynamic inference and prediction at the right timescales. So it's not that we're co-opting a system that usually is intended to move our arms. Instead, it's that system is for making predictions. And some of those predictions come true and are proprioceptive predictions and create movement, and other predictions are purely perceptual predictions. And also because humans use auditory feedback timing to inform or guide movement, which is why this particular type of, this branch of the predictive processing is so strong there. And there's more to say about that, but I don't have time to say it. So thank you so much for your attention. We've got Dr. Ani Patel and I have our TICS paper with these testable, with these hypotheses and make some testable predictions. And so if you're interested in that, you can check that out. And I've got this bioRxiv paper I mentioned up. And I would love to take some questions and thoughts.
Awesome. Thank you so much, Jonathan. I'll just quickly say, because I know some people might have to go right at 11, I'll just announce that our next journal club will be Dr. Jim Hayes of the Department of Neurobiology at the University of Utah, and that'll be on Tuesday, March 23rd at 10 a.m. And I'll put it in the chat. But thank you so much for that awesome talk, and we can take some questions. So I see that Ree has their hand up.
Yes, I'm Ria. Hi, thank you very much for your nice talk. I have one question about prediction and basal ganglia, because there is work by Mattel and Meck about striatal beat frequency model, and they say substantia nigra pars compacta dopamine neurons are firing, as function of prediction of when the reward is coming. I'm very curious why you didn't incorporate this part of prediction and prediction error, like the reward prediction error in terms of timing and path compactor. So why you didn't incorporate this part in your hypothesis?
So the dopaminergic cueing aspect of the hypothesis that we were talking about, I think that lines up reasonably well with the questions of expected reward time. This is from the substantia nigra pars compacta. And so I guess in our picture, the expectation of the auditory input of a beat is then, and the learned expectation of that is what's modulating that dopaminergic signal. So we don't have it neatly lined up with reward prediction error at this point. But I think that some recent dopaminergic literature, there's a fantastic paper, Believing in Dopamine by Gershman and folks, that is starting to bring together the idea of reward prediction error and dopamine used to modulate action. And so we're sort of still trying to work through what the relationship is there. But I guess more broadly for the striatal beat frequency model, I think there's a relationship here where that model is saying that the state of things like SMA, motor regions, is then detected by the striatum to indicate timing. And we're sort of trying to close that loop and say, like, okay, but the state in striatum is also instructing those timekeepers in cortex on what to do. So there's a relationship between this model and the striatal beat frequency model, but there's some important distinctions.
Okay, thank you.
Awesome. I see Jessica Gran has her hand up.
Yes, thank you. Thank you for a really nice talk, Jonathan. You've got a lot there to get through, and that was really clearly presented. Well done.
Thank you.
My question is probably where you left off, actually. So a lot of the data that you're presenting actually comes from non-human animals about the roles of the SMA and the basal ganglia. And in terms of a motor control perspective, the theories about what these areas do don't tend to differ very much for humans and non-human animals. So what do you think about that? I mean, obviously it makes implications that we should see this in non-human animals if the machinery is relatively conserved, and people have different opinions about that. So I just wanted to know your thoughts.
Yeah, I mean, I think that I'm sort of torn between two different hypotheses. I've got a bit of a slide on that here, just in case somebody asked that question. I think that the overarching idea for why it would be different in humans is that humans are using auditory timing and precise auditory timing to inform our estimates of where we are in movement much more than other animals. I imagine that other animals are going to be using the precise timing of footfalls, for instance, somatosensory input to inform their estimates of where they are in movement. And so we might see something more similar to beat-based timing in animals through somatosensation. But in humans, we've got two things going on. We've got vocal learning, which goes with this whole vocal learning hypothesis that Anya has helped map out. And we've also got bipedalism, where some of the best input we can get from about what our feet are is when we hear them touch the ground. And the precise timing is going to be really important for us not falling over. And so I think that some combination of those things may be the reason that we see these beat-based timing behaviors so much more developed in humans than in animals.
Awesome, thanks.
Jennifer Kula has her hand up, and then maybe we will wrap up there.
Hi, I'd just like to thank both speakers for a couple of really interesting talks. So you concentrated on beat-based timing and auditory input, but of course the overlap between the motor system and timing regions of the brain has been noticed a whole load of different timing areas. And I just wondered how applicable or how ubiquitous your model was for other areas of timing. So timing single durations or timing visual stimuli and so on. Have you thought about those?
So for short durations, I think that the key thing that is making that makes beat-based timing different is just the fact that expected input or real input at the end of one duration is then the cue to start timing the next one. And that cue is moderated by basal ganglia. But SMA doing that movement tracking, I think, is more of a general timing mechanism. And for shorter durations, I would imagine that SMA is similarly involved in interval timing. It just doesn't need the same sort of, when it reaches this point, that cues it to go again, that the basal ganglia offers. And right, so I think that, and the mathematical model that I mentioned on bioRxiv, I think would apply to both situations, although for longer durations, you need some way for these short chunks of activity in SMA to then turn into these longer arcs, which then may involve some kind of basal ganglia sequencing. Yeah, I think this is very relevant to questions of non-beat-based timing as well.
Okay, thanks.
So thank you, Jonathan, and thank you everyone for attending. That was an
awesome journal club today. Very, very interesting. Thanks to Austin and Jonathan and to the ECR committee for organizing. And just so you know, our next journal club, like I said before, will be Dr. Jim Hayes of the Department of Neurobiology at the University of Utah on Tuesday, March 23rd at 10 a.m. So put that on your calendar. So 10 a.m. Eastern time. Sorry, I'm very egocentric in my time zone right now. But I hope everyone enjoyed and can make it to the next one.
Thank you.